Hello, it's Mr. Singleton, one of the Associate Directors from Outward Academy Grange Trust. This lesson is all about enzymes and the properties of enzymes. So last lesson we looked at digestion and we touched on these enzymes and where they were made. This lesson just goes a little bit more in depth. The next lesson is really important. It's one of the required practicals about enzymes. So this is going to build up to that practical so you understand what's happening a little bit more. So this is a specification that AQA supplied. This is what can come up in the exams. So there's this lock and key theory, and then you must know the production of each enzyme that we're going to talk about. And those enzymes are amylase, protease, and lipase. And also in this, there's a bit about bile. Now I touched on bile in the previous lesson, so this lesson isn't going to go too much into that. What I want you to do for a starter is just recall the parts of the digestive system. So I want to know what numbers 1, 2, 3, 4, and 5 are in the digestive system. You can do this in the back of your book or on a whiteboard. Or if you're working by yourself, you can just think about this in your head, but try and come up with some answers. I'll give you 2 minutes. Off you go. You've got 1 more minute. Okay, so number 1 was the mouth. Number 2 was the esophagus, which is the pipe that takes food down into the stomach. Number 4 was the small intestine, which is actually really, really long. It's about 9 foot, in fact. But it's called the small intestine because of the diameter of the tube. And number 5 was the large intestine, which again is shorter than the small intestine, but has a larger diameter. Title for today is Properties of Enzymes. That needs to be in your book with today's date underlined. If you're not using books, you're working on your revision cards, then again, Properties of Enzymes for your title of your revision card. Please remember that this is for Biology Paper 1. So if I was you and I was making revision cards, Biology Paper 1 in the top corner, so you know which revision cards are for which exam that you're going to take. This one's usually taken in May. I'll give you a minute to get that sorted. The challenge today is to describe how enzymes work, and you need to give at least 3 examples of digestive enzymes. And then the aspire we're going to work on is the lock and key hypothesis, and then the conditions needed for enzymes to work. So we're going to talk a little bit about denaturing and things, and why bile is important then. So first off, what is an enzyme? Well, enzymes, you can say, are called biological catalysts, meaning they are made up of something that is found in the body, and they speed up reactions. What they are made up of is proteins, and they have this really specific shape, okay, and it's called an active site. And the active site is the part where everything happens, where they actually break down molecules. The molecule that they break down is given a specific word called a substrate, and that's what they break down. So in terms of the lock and key, only one type of enzyme can actually break down one type of molecule. So for example, let's just say protease, which is an enzyme, it can only break down proteins. It can't break down starch. Okay, so it's like the lock. A key can only open one type of lock. Another key can't open that lock. They're both specific, which is why we use that idea for an enzyme as well, because they are specific. What I've done is I've just recorded a little video just so you can see this drawn out a little bit, just to explain it. So next slide. So I'm just going to talk to you about enzymes and the structure of enzymes. It's probably easier to draw this than try and describe it through the PowerPoint picture. So look, here's our protein, which is the enzyme. And what happens is we usually draw it with this little bit here. So this little bit here that I've got here, and you have to excuse this writing a little bit. This bit is called the active site. Okay, and this is the key bit of this enzyme, because anything that's going to be broken down by this enzyme is going to fit into this active site. And what we call them is substrates. So this enzyme has a very specific shape, and it can only break down this substrate. So let's say this enzyme is amylase, then this substrate is the only one, which will be starch, which can fit into this active site. Okay, if a protein molecule comes along, let's get rid of that starch molecule. If a protein molecule comes along and has a different shape, because proteins have a different shape, let's say it's like a hexagon. Now this cannot fit into this active site, and if it can't fit into that active site, then the enzyme can't break it down. So we say this enzyme is specific. It can only break down certain things. So amylase can only break down starch, protease can only break down proteins. And that's because this active site is a certain shape, and no other different molecules can fit inside that active site. The whole point of enzymes is that so we can get these really large molecules that we eat, and we can break them down into smaller ones, and they have to be small enough to pass through the cell membrane. So they have to be really soluble and much smaller, and then once they can get through the cell membrane, they can get into bloodstreams and get around your body. And the small molecules that we're looking for, really, the really important ones, are called amino acids and glucose. So I just want you to have a little think about what those might be used for. So where have you come across glucose and how is it used in the body? I think you probably know that one more than the amino acids. But definitely by the end of this lesson, you'll know what amino acids make up. You might have a bit more of a clue. So why do we need glucose and why do we need amino acids? I'll give you 15 seconds to have a quick think about it. Why are they important in the body? Okay, so glucose is vital for respiration, and that respiration releases energy in the cells so that you can move, you can grow, you can do all the things that you need to do. And amino acids are really important to make new proteins. And proteins make up enzymes, hormones, they let your cells grow, they let your cells repair, so they are vital in life. So we talked a little bit about the 3 different types of enzymes that you need to know. So these are the ones that AQA want you to know. for your exams. One is called protease. Now, if you look at those blue arrows in the middle here, protease, look how it's spelled, P-R-O-T, and that breaks down protein. Carbohydrase breaks down carbohydrates, and lipase. Now, I've spoken through all of this. I would call fats lipids. So lipase breaks down lipids or fats. They are both the same thing. You also need to know what they break those molecules down into. So you have to think a little bit. Protein is this really long molecule that's just made up of amino acids. So when it's broken down, it just makes amino acids. Carbohydrates are made up of sugar. Now, starch is an example of a carbohydrate, and that is made up of glucose. So starch is just a long molecule of glucose, and when it breaks down by carbohydrates, it makes glucose. Fat is the one that most students get wrong or forget one part. So a lipid is made up, or a fat is made up of fatty acids and glycerol. They are two different parts. And when a lipase enzyme breaks down a fat or a lipid, it makes fatty acids and glycerol. I'm making that really obvious that I keep on saying and glycerol. Most students get the fatty acids part, but forget glycerol. Please don't forget glycerol. I'm just going to give you an example of a carbohydrase enzyme because we talked about it in the last lesson. It's called amylase. So amylase is the enzyme that breaks down starch into glucose. It's just an example of one of those types of enzymes. So I'm going to keep going through these because they're really important and lots of times people forget these. So amylase is a type of carbohydrate and it's made in the pancreas. This is the other part that you need to know where each one is made. So it's made in the pancreas, the small intestines and the salivary glands, and it breaks starch down into sugars. Protease is made in the pancreas and the stomach and the small intestine. And you might come across these enzymes, pepsin and trypsin. It's a little bit more A level, but sometimes they talk about it in the exams. They are just examples of protease enzymes. So that's where they are made, in the stomach, small intestine and pancreas. And they will break down proteins into amino acids. And then the last one you need to know is lipase, and that's produced in the pancreas and the small intestine. And it breaks it down into fatty acids and glycerol. Now, if you are ever struggling with where are these enzymes made, look at each one. They are all made in the pancreas and they are all made in the small intestine, but just be aware that sometimes AQA will turn around and say, protease is made in the small intestine and pancreas, and which other place? So please be aware that you need to know all three, because inevitably they always choose the ones that you know where they're made, and then you have to pick up the third one. So that is the challenge done. We've described how enzymes work and we can give 3 examples of each enzyme. I want you to try a Google quiz now. So I'll give you 4 minutes to do your Google quiz. Off you go. You've got 2 more minutes left. Okay. So these are the types of equations you might come across. I'm not going to ask you to complete these, it's just so you're aware. So carbohydrates or starch is broken down by carbohydrates, and what we call the products, what's made, is the glucose. Protein is broken down by protease, and the products are amino acids. And then lipids and fats, or lipids or fats, sorry, is broken down by lipase, and the products are fatty acids and glycerol. So I want you to draw this table in your book: enzymes, where it's produced, the substrate, and then the products for each one of those. I'll give you 5 minutes because I want you to draw that out properly in your book, nice and neatly, or on your revision card. It's perfect for a revision card because it has everything you need. Off you go. So I'm going to give you a little bit of help with this. So amylase, I can help you out. It's produced in the mouth, and the substrate, the thing that it breaks down, is starch, and the product is glucose. Okay, you got 2 more minutes left. One more minute. Okay, let's go through these answers. So amylase, made in the salivary glands or the mouth, small intestine and pancreas, breaks down starch or the substrate starch and produces other products are sugar. Protease, the only way you would have got this is seeing that it was made in the stomach, and then you could have worked out that it was protease. Then it breaks down proteins, and the products are amino acids. And then lipase breaks down lipids made in the small intestine and pancreas and produces fatty acids and glycerol. So a quick Google quiz. There's only a couple of questions. I'll give you 3 minutes to complete those. Off you go. Two more minutes. Okay. So the last part of this is about the conditions that help enzymes to work and why we have bile, etc. So really extreme conditions will cause the enzymes to change shape, and that means they denature. We give that word denature to them. They don't die because they were never classed as a living thing. So denaturing is when the active site changes shape, and once the active site changes shape, that molecule that it's going to break down can't fit in there, and the enzyme is now absolutely useless to us. So denaturing is just when the active site changes shape. Once the active site changes shape, then the substrate can't break down, and now the enzyme's pointless to us. So there's two bits. Is pH, and that is just talking about how acidic the environment is for the enzyme to work in. So these are two different types of enzymes, and pepsin is quite unique really, because pepsin works best in pH 2, so in a very acidic condition. Now where is that in your body? Well, it's in your stomach. So pepsin is found in your stomach. It's a protease enzyme, and it works really well in that low pH. However, amylase, which is found in your mouth, it does not work well at pH 2 at all. It would denature and it would not work, but it works really well in pH 7 conditions, such as your mouth. So that works quite well actually, because lots of your enzymes, which are found in microbes and things, once they go into your stomach, the enzyme stops being able to work, and the microbe won't work anymore. So that's really good, but then proteins can still be broken down. The other thing which can denature enzymes is temperature. So most enzymes will work the best at 40 degrees Celsius, and it's not by luck that suddenly this has happened. Our body is at 40 degrees Celsius, or kept at around 37 degrees Celsius, because that's when enzymes work best. If they didn't work best at that temperature, then our body temperature would not need to be there. I'm talking about enzymes, not just digestive enzymes, but enzymes that control your respiration. Okay, they work best at 40 degrees Celsius. If your enzymes got too high, the temperature got too high, then those enzymes can't work and you would die. So if you look at this here, if you go above, let's say 50 degrees Celsius, your enzymes stop working. They have become denatured and they won't work anymore. This is why it's really important to keep your body temperature around 37 degrees Celsius, because it stops your enzymes from denaturing and it makes them work the best. Now lower down the temperature, look between 10 and 30, the enzyme still works a little bit, but not very well. The enzyme hasn't denatured at this temperature. All that's happened is that the molecules, the enzyme, and the thing that it's going to break down does not have very much kinetic energy, so it just doesn't work as well. So that is how enzymes work and examples of enzymes, and we've talked about the conditions for enzymes to work as well. I'm just going to talk a little bit more about denaturing. So what actually happens is, as you get too hot, what happens is the enzymes break down because you are breaking bonds in the active site, and the active site changes shape. And the same thing happens with pH. As the pH gets too low, it affects the active site. The bonds in the active site start to break, and then the active site changes shape, and that's why it denatures. If the pH gets too high, it becomes very alkali. Similar thing, bonds break, changes the active site, and that denatures the enzyme. So when you try a Google quiz on these bits, I'll give you 4 minutes, off you go. Okay, you've got 2 more minutes. You've got 1 more minute. So if you're making revision cards, I still suggest you doing this. Make sure you put biology paper 1 in the top corner, just so you know which exam this is for. And I would title it Enzymes. So usually, either or, AQA will ask either what is an enzyme or what is a hormone. So enzyme, 3 mark question. Proteins, one will give you 1 mark, which have an active site, which will give you 2 mark. And they are specific, or you can say they speed up reactions. They will give you the marks for that. Digestive enzymes are those ones which break down molecules. There are 3 that you need to be aware of. So amylase or carbohydrates. Amylase breaks down starch into glucose, and it's found in the small intestine and saliva. Lipase breaks down lipids into fatty acids and glycerol, and found in the small intestine. Protease breaks down proteins into amino acids, found in the stomach, and all of these digestive enzymes are made in the pancreas. The next bit of the revision card is about denaturing, so I'll give you 2-3 minutes to write that bit down. If you're struggling, pause, obviously. Off you go. So next revision card, if you haven't finished again, pause it. This revision card is just about denaturing. So denaturing occurs when the active site changes shape, so molecules will no longer fit into the active site. And the two conditions which can denature enzymes are pH and temperature. Now, a low temperature just means that the enzymes and the molecules don't have enough kinetic energy to react. They won't actually denature the enzyme. So if you go into a very, very cold environment, the reason why you get hypothermia and things like that is because the enzymes can't react quick enough with the molecules because they don't have enough kinetic energy. But if you go into a high temperature, then the bonds break in that enzyme and the active site changes shape, so then it denatures it. pH also affects the active site, so therefore then will denature the enzyme. And you've got to remember that the stomach has an acid in it, hydrochloric acid, and that generally denatures enzymes. However, it does have a special enzyme, protease, that works at low pHs. I'll give you 2 minutes to write those bits down, and then we'll just talk about one little last bit and then get on with exam questions. Okay, you got 1 more minute. Right, so if you haven't got this down and you're trying to get this down, then just pause the video and then you can get it sorted. So on to the last bit, exam questions. Now just a couple of hints here. You know, if they're talking about something breaks down protein, then that makes amino acids, the pH is going to get lower and it's going to become more acidic. And that's the same with lipids. If we are breaking down lipids, that solution gets more acidic, so it goes to a lower pH. So just be aware of that when you're looking at some exam questions, and that's about applying your knowledge a bit. If it says amino acids at it, that's going to lower the pH. If it says fatty acids, that's going to lower the pH of the solution. Just a couple of helpful hints for you. Well done. If you're still going on with this, super, well done. There are some exam questions there for you. Either do them on the Google Doc or you can print them off and do them on paper and hand them in to your teacher. Just get some exam question practice. It'd be really useful for you.